Subscribe to Boring Science for sleep for new videos every day. Now, let us begin. The movement of water through freshwater wetland ecosystems is a process that has been studied by hydrologists, ecologists, and environmental scientists for well over a century. And yet the full complexity of these movements continues to be examined in ever greater detail with each passing decade. Wetlands, broadly defined as areas where the soil is saturated with water for a significant portion of the year, occupy an estimated 6% of the Earth's land surface, which amounts to roughly 860 million hectares when one accounts for the various types of freshwater marshes, swamps, bogs, and fens that exist across every continent except Antarctica. The water that flows into, through, and out of these ecosystems follows pathways that are remarkably intricate, involving interactions between precipitation, surface runoff, groundwater seepage, evaporation, and transpiration from the dense vegetation that characterizes these habitats. To begin to understand the water cycle in freshwater wetlands, it is first necessary to consider the sources of water that feed these systems. Precipitation, in its various forms including rain, snow, sleet, and hail, represents the most direct input of water to any wetland. In temperate regions, the average annual precipitation that falls upon a typical freshwater marsh might range from 600 to 1,200 millimeters per year, depending on the specific geographic location and prevailing climatic conditions. In tropical wetlands, such as those found in the Amazon basin or the Pantanal region of South America, annual precipitation can exceed 1,600 millimeters, creating conditions of nearly perpetual saturation. The rain that falls directly onto the surface of a wetland is immediately incorporated into the standing water or absorbed into the upper layers of the soil, where it begins its slow journey through the substrate. Surface runoff constitutes another major source of water for freshwater wetlands. When rainfall exceeds the infiltration capacity of the surrounding upland soils, the excess water flows downhill under the influence of gravity, following the contours of the landscape until it reaches the lowest available point, which is often a wetland depression. The volume of surface runoff that enters a wetland is determined by a number of factors, including the intensity and duration of the rainfall event, the slope of the surrounding terrain, the permeability of the upland soils, and the extent to which the landscape has been modified by human activities such as agriculture, urbanization, or deforestation. In a natural, undisturbed landscape, the proportion of rainfall that becomes surface runoff is typically between 10 and 30%, though this figure can be considerably higher in areas where impervious surfaces such as roads, parking lots, and buildings prevent water from soaking into the ground. Groundwater represents a third and particularly important source of water for many freshwater wetlands. The relationship between wetlands and groundwater is complex and bidirectional, meaning that wetlands can both receive water from and contribute water to the underlying aquifer. In areas where the water table intersects the land surface, groundwater seeps upward into the wetland, providing a steady and often year-round supply of water that helps to maintain saturated conditions even during periods of low rainfall. This upward movement of groundwater, known as groundwater discharge, is driven by differences in hydraulic head between the aquifer and the wetland surface. The rate of groundwater discharge into a wetland depends on the hydraulic conductivity of the subsurface materials, the gradient of the water table, and the depth to the water table in the surrounding landscape. The hydraulic conductivity of soils and sediments beneath a wetland varies enormously depending on the texture and composition of these materials. Sandy soils, which have relatively large pore spaces between their constituent grains, can have hydraulic conductivities ranging from 1 to 100 meters per day, allowing water to flow through them with relative ease. Clay soils, by contrast, have extremely small pore spaces and correspondingly low hydraulic conductivities, often measuring less than 1/100th of a meter per day. The sediments that accumulate at the bottom of a wetland are typically a mixture of organic matter and mineral particles, and the hydraulic conductivity of this mixture can range from approximately 0.01 to 10 meters per day, depending on the degree of decomposition of the organic material and the proportion of fine mineral particles present. Once water enters a freshwater wetland, whether from precipitation, surface runoff, or groundwater discharge, it begins to move through the system in a variety of ways. The most visible movement is the flow of surface water across the wetland, which is driven by differences in water level between the inlet and outlet of the system. In a typical freshwater marsh, the depth of standing water might range from just a few centimeters to over a meter, and the velocity of surface flow is generally quite slow, often measuring less than 1 centimeter per second. This sluggish movement of water is one of the defining characteristics of wetlands and is responsible for many of their ecological functions, including the trapping of sediments, the removal of nutrients and pollutants, and the provision of habitat for a wide variety of plant and animal species. The flow of water through a wetland is significantly influenced by the presence of vegetation, which creates resistance to flow and causes the water to spread out across the wetland surface rather than channeling into narrow, fast-moving streams. The stems, leaves, and root systems of wetland plants act as a natural filter, slowing the movement of water and allowing suspended particles to settle out of the water column. Research conducted at various wetland sites around the world has shown that the presence of dense vegetation can reduce flow velocities by as much as 90% compared to open water conditions. The Manning's roughness coefficient, which is a measure of the resistance to flow exerted by the bed and banks of a channel, typically ranges from 0.03 to 0.15 in open channels, but can exceed 0.3 in densely vegetated wetlands, reflecting the enormous drag that plant matter exerts on the flowing water. Evaporation and transpiration, collectively referred to as evapotranspiration, represent the primary pathways by which water leaves a freshwater wetland and returns to the atmosphere. Evaporation occurs when water molecules at the surface of the standing water or saturated soil gain enough energy from solar radiation and atmospheric heat to escape into the air as water vapor. The rate of evaporation from a wetland surface is influenced by a number of factors, including air temperature, humidity, wind speed, and the amount of solar radiation reaching the surface. In temperate climates, evaporation rates from wetland surfaces typically range from 3 to 6 millimeters per day during the summer months and may drop to less than 1 millimeter per day during winter. In tropical regions, evaporation can proceed at rates of 5 to 8 millimeters per day throughout much of the year. Transpiration, the process by which water is absorbed by plant roots, transported upward through the stem, and released as vapor through tiny pores called stomata on the surfaces of leaves, is an equally important pathway for water loss from wetlands. The rate of transpiration depends on the species of plant, the density of the vegetation, the availability of water in the root zone, and atmospheric conditions such as temperature, humidity, and wind speed. A single mature willow tree growing in a wetland environment can transpire as much as 400 liters of water per day during the growing season. And a dense stand of cattails or reeds can transpire water at rates comparable to or even exceeding the rate of evaporation from an open water surface of the same area. Measurements taken at research wetlands in the United States and Europe have shown that total evapotranspiration from vegetated wetlands can range from 500 to 1,200 millimeters per year, which in some cases approaches or exceeds the amount of precipitation received. The balance between water inputs and water outputs in a freshwater wetland is described by the wetland water budget, which is a fundamental concept in wetland hydrology. The water budget can be expressed as a simple equation. The change in water storage within the wetland over a given period equals the total inputs minus the total outputs. Inputs include precipitation falling directly on the wetland, surface runoff entering from the surrounding catchment, and groundwater discharge from the underlying aquifer. Outputs include evapotranspiration, surface outflow from the wetland, and groundwater recharge, which occurs when water from the wetland seeps downward into the aquifer. If the inputs exceed the outputs, the water level in the wetland rises. If the outputs exceed the inputs, the water level falls. Over the course of a year, most natural wetlands maintain a rough balance between inputs and outputs, though significant seasonal fluctuations are common. The concept of hydroperiod is closely related to the water budget and refers to the pattern of water level fluctuations that a wetland experiences over the course of a year. The hydroperiod encompasses both the duration of inundation, which is the length of time that standing water is present, and the depth of inundation, which is the maximum depth of standing water that occurs during the wettest period. Different types of freshwater wetlands have characteristically different hydroperiods. Permanent marshes, which are found in areas with high water tables or abundant surface water inputs, maintain standing water throughout the year and have hydroperiods of 12 months. Seasonal marshes, which are typical of many temperate regions, may be inundated for 6 to 9 months of the year, with water levels dropping below the soil surface during the driest months. Ephemeral wetlands, such as prairie potholes and vernal pools, may hold water for only a few weeks or months following snowmelt or heavy rainfall events. The hydroperiod of a wetland is one of the most important factors determining the types of plants and animals that can inhabit the system. Plants that grow in permanently flooded wetlands must possess adaptations that allow them to tolerate continuous submersion of their root systems in oxygen-depleted soils. These adaptations include the development of parenchyma, which is specialized tissue containing large air spaces that facilitate the transport of oxygen from the aerial portions of the plant down to the submerged roots. Many wetland plants also possess the ability to carry out anaerobic respiration in their root cells, allowing them to generate energy even when oxygen is scarce. The common cattail, Typha latifolia, is an example of a plant that is superbly adapted to permanently or semi-permanently flooded conditions. Possessing extensive parenchyma in its stems and rhizomes and capable of growing in water depths of up to 150 centimeters. The soils that develop in freshwater wetlands are distinctly different from those found in upland environments, and these differences are largely a consequence of the prolonged saturation that characterizes wetland hydrology. When soil is saturated with water for extended periods, the oxygen that is normally present in the pore spaces between soil particles is consumed by microbial respiration and cannot be replenished by diffusion from the atmosphere because the pore spaces are filled with water rather than air. This creates anaerobic or oxygen-free conditions in the soil, which profoundly affects the chemical and biological processes that occur within it. Under anaerobic conditions, certain chemical elements, most notably iron and manganese, undergo reduction reactions that change their oxidation state and alter their solubility. Ferric iron, which has an oxidation state of +3, is reduced to ferrous iron, with an oxidation state of +2, becoming much more soluble in the process. This reduction of iron gives wetland soils their characteristic gray or bluish-gray color, a feature that soil scientists refer to as glaying. The accumulation of organic matter is another distinctive feature of wetland soils. In upland soils, dead plant material is rapidly decomposed by aerobic microorganisms, and relatively little organic matter accumulates over time. In wetlands, however, the anaerobic conditions that prevail in the saturated zone greatly slow the rate of decomposition, allowing organic matter to accumulate year after year. Over centuries and millennia, this accumulation can result in the formation of thick deposits of peat, which is partially decomposed plant material that retains much of the original structure and chemical composition of the living plants from which it was derived. Peatlands, which are wetlands where peat has accumulated to a depth of at least 30 to 40 centimeters, cover an estimated 400 million hectares globally, with the majority located in the boreal and subarctic regions of Canada, Russia, and Scandinavia. The peat deposits in some of these northern wetlands can be several meters thick and may have taken thousands of years to form, with typical accumulation rates of approximately 1 millimeter per year. The water-holding capacity of peat is truly remarkable, and this property has significant implications for the hydrology of peatlands. A cubic meter of peat in its natural, undrained state can hold between 800 and 900 liters of water, which is several times more than the same volume of mineral soil. This enormous water-holding capacity means that peatlands act as vast natural reservoirs, storing water during wet periods and releasing it slowly during dry periods. The total volume of water stored in the world's peatlands has been estimated at between 500 and 600 cubic kilometers, which is comparable to the volume of water in some of the world's largest freshwater lakes. This stored water plays an important role in regulating stream flow in the catchments where peatlands are located, moderating the peaks and troughs of the hydrograph, and providing a sustained base flow to rivers and streams during periods of low rainfall. The movement of water through peat is governed by the same physical principles that govern the flow of water through any porous medium, but the unique properties of peat result in some distinctive hydrological behaviors. The hydraulic conductivity of peat varies enormously with depth, generally decreasing from relatively high values near the surface, where the peat is relatively uncompacted and retains much of its original fibrous structure, to very low values at greater depths, where centuries of compression have reduced the pore spaces to a fraction of their original size. Near the surface of a typical sphagnum peatland, the hydraulic conductivity might be in the range of 10 to 100 meters per day, which is comparable to that of a sandy soil. At depths of 1 to 2 meters, however, the hydraulic conductivity may have decreased to less than 0.01 meters per day, which is more comparable to that of a clay soil. This vertical gradient in hydraulic conductivity has important consequences for the flow patterns within the peatland, causing the majority of water movement to occur in the upper, more permeable layers, while the deeper layers remain relatively stagnant. The interaction between surface water and groundwater in freshwater wetlands is a subject that has received increasing attention from hydrologists in recent decades, as improvements in measurement technology and computational modeling have made it possible to study these interactions in greater detail than was previously feasible. One of the key findings of this research is that the direction and magnitude of water exchange between a wetland and the underlying aquifer can vary significantly over both space and time. A single wetland may simultaneously receive groundwater discharge in one area and lose water to groundwater recharge in another. Depending on local variations in the topography of the water table and the hydraulic properties of the underlying sediments. Similarly, the same location within a wetland may experience groundwater discharge during the wet season, when the water table in the surrounding landscape is high, and groundwater recharge during the dry season, when the water table has fallen below the level of the wetland surface. The chemical composition of the water in a freshwater wetland reflects the various sources from which it is derived and the biogeochemical processes that occur within the wetland itself. Precipitation, which is the purest natural source of water, typically has a very low concentration of dissolved solids, usually less than 10 milligrams per liter. Surface runoff, which picks up dissolved and particulate materials as it flows across the landscape, generally has higher concentrations of dissolved solids, often ranging from 50 to several hundred milligrams per liter, depending on the geology and land use of the catchment. Groundwater, which has been in contact with subsurface minerals for extended periods, tends to have the highest concentrations of dissolved solids, with values commonly ranging from 100 to over 1,000 milligrams per liter. The relative contributions of these different water sources to a wetland therefore have a strong influence on the overall water chemistry of the system. Within the wetland itself, a complex array of biogeochemical processes further modifies the chemistry of the water. Photosynthesis by aquatic plants and algae removes dissolved carbon dioxide from the water, raising the pH and reducing the concentration of dissolved inorganic carbon. Respiration by plants, animals, and microorganisms has the opposite effect, releasing carbon dioxide into the water and lowering the pH. The decomposition of organic matter under anaerobic conditions produces a variety of reduced chemical species, including ferrous iron, manganese manganese, ammonium, and hydrogen sulfide, as well as methane, which is a potent greenhouse gas. The production of methane in wetlands is a globally significant process, with freshwater wetlands estimated to contribute between 100 and 200 teragrams of methane to the atmosphere each year, which represents approximately 20 to 30% of all methane emissions from natural sources. The nutrient cycling that occurs within freshwater wetlands is intimately linked to the water cycle, as the movement of water through the system transports dissolved nutrients from one location to another and creates the chemical conditions that determine whether nutrients are retained within the wetland or exported to downstream water bodies. Nitrogen and phosphorus are the two nutrients of greatest concern in wetland biogeochemistry, both because they are essential for plant growth and because their excessive enrichment of surface waters can lead to eutrophication, a process in which algal blooms and other nuisance organisms proliferate, depleting dissolved oxygen and degrading water quality. Wetlands have long been recognized for their ability to remove nitrogen and phosphorus from water that passes through them, and this capability has been exploited in the design of constructed wetlands, which are engineered systems that mimic the nutrient removal processes of natural wetlands to treat municipal wastewater, agricultural runoff, and industrial effluent. The removal of nitrogen in wetlands occurs primarily through the process of denitrification, in which specialized bacteria convert nitrate, a dissolved form of nitrogen, into nitrogen gas, which escapes to the atmosphere. Denitrification requires anaerobic conditions, a supply of organic carbon to serve as an electron donor, and a supply of nitrate to serve as an electron acceptor. All three of these requirements are typically met in the saturated zone of a wetland, where organic matter is abundant, oxygen is scarce, and nitrate is supplied by the incoming water. The rate of denitrification in wetlands has been measured at numerous sites around the world and typically ranges from 50 to 300 milligrams of nitrogen per square meter per day during the growing season. though rates as high as 600 milligrams per square meter per day have been recorded in wetlands receiving high loads of nitrogen-rich wastewater. The phosphorus cycle in freshwater wetlands operates through mechanisms that are fundamentally different from those governing the nitrogen cycle, and understanding these differences is essential for appreciating the role that wetlands play in maintaining water quality in the broader landscape. Unlike nitrogen, which can be permanently removed from the aquatic system through conversion to a gaseous form, phosphorus has no significant gaseous phase and therefore cannot be removed from the wetland through volatilization. Instead, phosphorus removal in wetlands relies primarily on physical and chemical processes that immobilize phosphorus in the soil and sediments, preventing it from being transported downstream. The most important of these processes include sedimentation, in which phosphorus attached to suspended particles settles out of the water column and is deposited on the wetland floor, adsorption, in which dissolved phosphorus binds to the surfaces of soil minerals, particularly iron and aluminium oxides, and biological uptake, in which plants and microorganisms incorporate dissolved phosphorus into their tissues as they grow. The effectiveness of a wetland in removing phosphorus from the water that passes through it depends on a number of factors, including the concentration of phosphorus in the incoming water, the hydraulic loading rate, which is the volume of water entering the wetland per unit area per unit time, and the chemical properties of the wetland soil. Wetlands with soils that are rich in iron and aluminium oxides tend to be more effective at removing phosphorus because these minerals have a high capacity for absorbing dissolved phosphorus. The phosphorus adsorption capacity of a wetland soil can be determined by conducting laboratory experiments in which soil samples are exposed to solutions of known phosphorus concentration and the amount of phosphorus adsorbed is measured. These experiments have shown that the maximum phosphorus adsorption capacity of wetland soils typically ranges from 500 to 2,000 milligrams of phosphorus per kilogram of soil, though values outside this range are not uncommon. The seasonal patterns of water flow through freshwater wetlands are among the most important determinants of wetland ecology, and these patterns are, in turn, driven by the seasonal variations in precipitation, temperature, and evapotranspiration that characterize different climatic regions. In temperate regions of the northern hemisphere, the water cycle in freshwater wetlands follows a broadly predictable annual pattern. During the winter months, from approximately December through February, precipitation often falls as snow, which accumulates on the surface of the wetland and in the surrounding catchment. Evapotranspiration is minimal during this period because low temperatures and short day lengths limit both evaporation and plant transpiration. As a result, the water stored in the wetland may gradually increase over the winter, though in cold climates the surface of the wetland may be frozen, preventing the inflow of surface runoff. The spring thaw, which typically occurs between March and May in the northern hemisphere, marks a dramatic shift in the wetland water cycle. As temperatures rise and snow begins to melt, large volumes of meltwater are released over a relatively short period, often just a few weeks. This pulse of snowmelt runoff is typically the single largest input of water to the wetland during the entire year and can cause water levels to rise rapidly, sometimes by several tens of centimeters in just a few days. The volume of snowmelt runoff that enters a wetland depends on the depth of the snowpack, the rate at which it melts, and the proportion of the meltwater that reaches the wetland rather than infiltrating into the still partially frozen ground. In the prairie pothole region of central North America, where millions of small, shallow wetlands are scattered across a landscape of gently rolling glacial terrain, snowmelt runoff can account for as much as 70 to 80% of the total annual water input to these systems. Following the spring thaw, the wetland water cycle enters a phase dominated by evapotranspiration. As temperatures continue to rise and day lengths increase through the spring and summer months, the rate of water loss from the wetland through evaporation and transpiration accelerates markedly. By midsummer, evapotranspiration rates can exceed the rate of water input from precipitation, causing water levels to decline gradually over the course of the growing season. In many seasonal wetlands, this decline in water levels continues until the standing water disappears entirely, exposing the underlying mud and organic sediments to the atmosphere. The timing and duration of this drawdown period vary from year to year, depending on the amount of precipitation received during the growing season and the temperatures experienced, which in turn influence evapotranspiration rates. The autumn months bring a gradual reversal of the summer pattern. As temperatures decline, evapotranspiration rates decrease and precipitation begins to exceed water losses. Water levels in the wetland gradually recover, and by late autumn, many wetlands have returned to a state of partial or complete inundation. This seasonal cycle of flooding, drawdown, and reflooding is a fundamental characteristic of many freshwater wetlands and plays a crucial role in maintaining their ecological diversity. The alternation between wet and dry conditions creates a mosaic of habitats that can support a wider range of species than would be possible under conditions of permanent flooding or permanent dryness. The vegetation of freshwater wetlands is both a product of and a contributor to the water cycle, and the interactions between plants and water in these systems are remarkably complex. The root systems of wetland plants play a particularly important role in the movement of water through the shallow subsurface. As roots grow through the soil, they create channels and macropores that can significantly increase the hydraulic conductivity of the root zone, allowing water to flow more freely through areas of dense root development than through surrounding soils. When roots die and decompose, the channels they created may persist for months or even years, continuing to facilitate water movement long after the original root has disappeared. Studies using tracer dyes and other techniques have shown that preferential flow through root channels and other macropores can account for a substantial proportion of the total water movement through the upper layers of wetland soils, particularly during high-flow events when the pore spaces in the soil matrix are already fully saturated. The canopy of emergent wetland vegetation, which consists of the stems and leaves that extend above the water surface, also influences the water cycle in several important ways. The canopy intercepts a portion of the rainfall that would otherwise fall directly onto the water surface or soil. This intercepted precipitation is temporarily held on the surfaces of leaves and stems, where some of it evaporates back into the atmosphere without ever reaching the wetland surface. The proportion of rainfall that is intercepted by the canopy depends on the density of the vegetation, the size and shape of the leaves, and the intensity and duration of the rainfall event. In dense stands of emergent vegetation such as Phragmites australis, canopy interception can account for 10 to 20% of the total rainfall during small, low-intensity events, though the proportion decreases during heavy rainstorms when the storage capacity of the leaf surfaces is quickly exceeded. The transpiration of water by wetland plants is a process that is regulated by a complex set of physiological and environmental factors. The stomata, which are the microscopic pores on the leaf surfaces through which water vapor escapes, can open and close in response to changes in light intensity, humidity, temperature, and the water status of the plant. When conditions are favorable for photosynthesis, the stomata open to allow carbon dioxide to enter the leaf, but this opening simultaneously allows water vapor to escape. The rate of transpiration is therefore closely coupled to the rate of photosynthesis, and both processes are strongly influenced by the amount of sunlight reaching the leaf surface. On a clear summer day, a stand of cattails in a temperate freshwater marsh might transpire water at a rate of 6 to 8 millimeters per day, while on an overcast day, the rate might be reduced to 3 to 4 millimeters per day. The phenomenon of nocturnal transpiration, in which wetland plants continue to lose water through their stomata during the night, has been documented in a number of wetland species and appears to be more common than was previously thought. Although the stomata of most plants close at night in response to the absence of light, they may not close completely, and the small residual opening can allow significant amounts of water to escape over the course of a long summer night. Measurements of sap flow in wetland trees and shrubs have shown that nocturnal transpiration can account for 5 to 15% of the total daily water loss, depending on the species and environmental conditions. The ecological significance of nocturnal transpiration in wetlands is still not fully understood, but it has been suggested that it may help to maintain the flow of nutrients from the soil to the leaves during the night, ensuring a continuous supply of mineral elements for growth. The concept of the wetland water table, which refers to the upper surface of the zone of saturation within the wetland soil, is central to understanding the hydrology of these systems. The position of the water table relative to the soil surface determines whether standing water is present on the surface of the wetland and, if so, to what depth. When the water table is above the soil surface, standing water is present, and the depth of standing water equals the height of the water table above the soil surface. When the water table is below the soil surface, no standing water is present, though the soil may still be saturated at depth. The position of the water table fluctuates continuously in response to changes in the balance between water inputs and outputs and these fluctuations can be measured using simple devices called piezometers, which consist of a narrow tube inserted vertically into the soil with its lower end open or screened to allow water to enter. The installation and monitoring of piezometer networks has been a standard technique in wetland hydrology since the mid-20th century, and the data obtained from these networks have greatly advanced the understanding of water movement through wetland soils. A typical piezometer installation in a research wetland might consist of several dozen individual piezometers arranged in transects across the wetland, with each piezometer screened at a different depth to allow the measurement of hydraulic head at various levels within the soil profile. By comparing the water levels in piezometers screened at different depths, hydrologists can determine the vertical direction of water movement at each location. If the water level in a shallow piezometer is higher than in a deeper one at the same location, this indicates that water is moving downward from the wetland into the underlying aquifer, a condition known as groundwater recharge. Conversely, if the water level in the deeper piezometer is higher, water is moving upward from the aquifer into the wetland, a condition known as groundwater discharge. The spatial variability of groundwater surface water interactions within a single wetland can be quite remarkable. Research conducted at a number of study sites has revealed that wetlands often contain distinct zones of groundwater discharge and groundwater recharge, sometimes separated by distances of only a few meters. These zones may be associated with variations in the topography of the wetland surface, the permeability of the underlying sediments, or the presence of buried geological features such as sand lenses or gravel layers that serve as preferential pathways for groundwater flow. The identification and mapping of these discharge and recharge zones is important for understanding the overall water budget of the wetland and for predicting how the wetland might respond to changes in land use, climate, or water management practices in the surrounding landscape. The role of freshwater wetlands in the broader hydrological cycle of a river catchment has been a subject of considerable debate among hydrologists and water resource managers. One of the most commonly cited functions of wetlands is their ability to attenuate floods by storing water during periods of high flow and releasing it gradually over the following days and weeks. This flood attenuation function is a direct consequence of the large storage capacity of wetlands and the resistance to flow exerted by wetland vegetation, both of which slow the passage of floodwaters through the system and reduce the peak discharge downstream. The effectiveness of a wetland in attenuating floods depends on its size relative to the catchment area, its position within the catchment, the volume of flood storage available at the time of the flood event, and the characteristics of the flood itself. Numerous studies have attempted to quantify the flood attenuation capacity of freshwater wetlands, and the results have been variable. Some studies have found that the presence of wetlands in a catchment can reduce peak flood discharges by as much as 60 to 80%, while others have found much smaller effects or even no significant effect at all. This variability is partly due to differences in the types and locations of wetlands studied and partly due to differences in the hydrological conditions prevailing at the time of the study. A wetland that is already full of water at the beginning of a flood event will have little additional storage capacity and may actually contribute to downstream flooding by passing incoming water through the system without significant delay. Conversely, a wetland that has been drawn down during a preceding dry period will have substantial available storage and can absorb a large proportion of the incoming floodwater before any outflow occurs. The base flow maintenance function of freshwater wetlands is perhaps less dramatic than their flood attenuation role, but is equally important for the health of downstream aquatic ecosystems. Base flow refers to the sustained flow of water in a river or stream during dry periods, when there is no direct contribution from surface runoff. In many catchments, base flow is maintained by the gradual release of water from storage in the soil, aquifer, and wetlands. Wetlands contribute to base flow by slowly releasing the water they have stored during wet periods, providing a sustained supply of water to downstream channels during times when other water sources have diminished. This function is particularly important in regions where dry seasons are prolonged and base flow is essential for maintaining aquatic habitats, supporting fish populations, and providing water for human uses such as irrigation and drinking water supply. The measurement of water flow rates in freshwater wetlands presents considerable practical challenges that have influenced the development of specialized techniques and instruments for this purpose. Unlike rivers and streams, where flow is concentrated in a well-defined channel and can be measured relatively easily using current meters or weirs, the flow of water in wetlands is diffuse, shallow, and distributed across a broad area. Conventional current meters, which require a minimum depth of water in which to operate, are often unusable in the shallow, vegetation-clogged waters of a typical wetland. Instead, hydrologists have developed alternative methods for measuring wetland flows, including the use of acoustic Doppler velocimeters, which can measure flow velocities in very shallow water, tracer dilution methods, in which a known quantity of a conservative tracer such as sodium chloride or rhodamine dye is released into the wetland, and its concentration is monitored at downstream locations to determine the rate of water movement, and the water budget method, in which the flow through the wetland is calculated as the residual of all other measured inputs and outputs. The thermal regime of freshwater wetlands, which describes the patterns of water temperature variation over time and space, is closely linked to the water cycle and has important implications for the biological processes that occur within these systems. The temperature of the water in a wetland is determined by the balance between heat inputs, which includes solar radiation, conduction from the atmosphere and soil, and the heat content of inflowing water, and heat outputs, which include radiation from the water surface, evaporation, and the heat content of outflowing water. During the summer months in temperate regions, the shallow water of a wetland can warm rapidly under the influence of solar radiation, with surface temperatures sometimes reaching 25 to 30 degrees Celsius on hot, sunny days. At night, The water cools by radiating heat back into the atmosphere, and temperatures may drop by 5 to 10 degrees Celsius, resulting in a pronounced diurnal temperature cycle. The thermal stratification of water in deeper wetland pools and ponds can have significant effects on the biogeochemical processes occurring in these areas. When the surface water is warmed by solar radiation, it becomes less dense than the cooler water below, creating a stable layered structure in which the warm, less dense water floats on top of the cool, denser water. This stratification can persist for days or weeks during calm, warm weather and effectively isolates the bottom water from contact with the atmosphere. As microorganisms in the bottom sediments consume the dissolved oxygen in this isolated layer, the water becomes progressively more anoxic, creating conditions that favor the production of methane, hydrogen sulfide, and other reduced compounds. When the stratification eventually breaks down, either through cooling of the surface water or mixing by wind, the accumulated gases and reduced compounds are released into the water column, sometimes causing dramatic but short-lived changes in water chemistry. The influence of climate change on the water cycle in freshwater wetlands is a topic of growing concern among scientists and environmental managers. Climate models project that many regions of the world will experience changes in the amount, timing, and intensity of precipitation over the coming decades, and these changes are likely to have profound effects on wetland hydrology. In some regions, such as the high latitudes of the northern hemisphere, precipitation is projected to increase, potentially leading to the expansion of existing wetlands and the creation of new ones. In other regions, particularly those in the subtropics and mid-latitudes, precipitation is projected to decrease, which could lead to the drying out of wetlands that have existed for centuries or millennia. Even in regions where total annual precipitation is not expected to change significantly, shifts in the seasonal distribution of precipitation could alter the hydroperiod of wetlands in ways that affect the plant and animal communities they support. Rising temperatures associated with climate change are expected to increase rates of evapotranspiration in most regions, which will tend to reduce the amount of water available to sustain wetlands. A temperature increase of 2 degrees Celsius, which is within the range projected by many climate models for the latter half of this century, could increase potential evapotranspiration by approximately 5 to 10% in temperate regions, and this increase could be sufficient to shift the water balance of some marginally wet areas from surplus to deficit, causing them to dry out. The melting of permafrost in Arctic and subarctic regions is another consequence of climate change that is already having dramatic effects on wetland hydrology in the far north. As permafrost thaws, the ice-rich ground collapses, creating new depressions that fill with water to form thermokarst lakes and wetlands. At the same time, the increased drainage through the now unfrozen ground can cause existing wetlands to lose water and shrink. The hydrological connectivity of freshwater wetlands, which refers to the degree to which water flows between a wetland and other water bodies in the landscape, is a concept that has important implications for both the ecological functioning of the wetland and its legal and regulatory status. A wetland that is directly connected to a river by a permanent channel is said to have high hydrological connectivity, while a wetland that is isolated from other water bodies and receives water only from direct precipitation and shallow groundwater seepage has low hydrological connectivity. The degree of connectivity can vary seasonally, with some wetlands being connected to adjacent water bodies only during periods of high water and becoming isolated during dry periods. Hydrological connectivity influences the movement of organisms, nutrients, and sediments between wetlands and other aquatic habitats and is therefore a key determinant of wetland biodiversity and ecosystem function. Wetlands with high connectivity tend to have more diverse fish communities because fish can move freely between the wetland and adjacent rivers or lakes, using the wetland as a spawning, nursery, or feeding habitat. These connected wetlands also tend to receive larger inputs of nutrients and sediments from upstream sources, which can support high levels of biological productivity, but can also lead to problems of eutrophication and sedimentation if nutrient loads are excessive. The classification of freshwater wetlands according to their hydrological characteristics has been a subject of study for many decades, and numerous classification systems have been proposed by scientists working in different countries and different wetland types. One of the most widely used classification systems in North America is the Cawarden system, which was developed by the United States Fish and Wildlife Service in 1979 and classifies wetlands based on their vegetation, soil type, and hydrological regime. Under the Cawarden system, freshwater wetlands are divided into two main categories: palustrine wetlands, which include marshes, swamps, bogs, and fens that are not associated with large lakes or rivers, and lacustrine and riverine wetlands, which are associated with lakes and rivers respectively. Each of these categories is further subdivided based on the dominant type of vegetation, the water regime, and other characteristics. The distinction between bogs and fens, two of the major types of peatland, is fundamentally hydrological in nature. Bogs are peatlands that receive all of their water from precipitation, with no significant input from surface runoff or groundwater. Because their only water source is rainwater, which has very low concentrations of dissolved minerals, bogs tend to be acidic and nutrient-poor environments that support a specialized flora dominated by sphagnum mosses, ericaceous shrubs such as blueberries and cranberries, and carnivorous plants such as sundews and pitcher plants. The pH of bog water typically ranges from 3.5 to 5, which is comparable to the acidity of vinegar, and the concentration of calcium, which is one of the most important mineral nutrients for plant growth, is usually less than 2 milligrams per liter. Fens, by contrast, receive a significant proportion of their water from groundwater that has been in contact with mineral-bearing rocks and soils, and as a result, fens tend to be less acidic and more nutrient-rich than bogs. The pH of fen water typically ranges from 5.5 to 8, depending on the mineral content of the groundwater, and calcium concentrations can range from 10 to over 100 milligrams per liter. The vegetation of fens is correspondingly more diverse than that of bogs, typically including a mixture of sedges, grasses, rushes, and herbaceous flowering plants, as well as some sphagnum mosses. In regions where calcareous groundwater is abundant, such as parts of the English Midlands, the Swiss Alps, and the Dinaric karst of southeastern Europe, fens may support particularly species-rich plant communities that include a number of rare and threatened species. The transition between a fen and a bog can occur naturally over the course of thousands of years through a process known as polutification, in which the progressive accumulation of peat gradually raises the surface of the wetland above the level of the surrounding water table, cutting off the supply of mineral-rich groundwater and causing the vegetation to shift from species adapted to mineral-rich conditions to those adapted to nutrient-poor, acidic conditions. This transition from minerotrophic conditions, in which the wetland receives mineral-rich water from the surrounding landscape, to ombrotrophic conditions, in which the wetland receives water only from precipitation, is one of the most well-documented examples of long-term ecological succession in wetland ecosystems, and has been studied extensively using paleoecological techniques such as pollen analysis and the examination of plant macrofossils preserved in peat cores. The microbial communities that inhabit the saturated soils of freshwater wetlands play a central role in the biogeochemical processes that are driven by the water cycle, and the diversity and activity of these communities are profoundly influenced by the hydrological conditions prevailing in the wetland. In the aerobic zone near the surface of the soil, where oxygen is available either from the atmosphere or from the roots of wetland plants, aerobic bacteria and fungi are the dominant decomposers of organic matter. These organisms break down complex organic molecules such as cellulose, lignin, and proteins into simpler compounds, releasing carbon dioxide, water, and mineral nutrients in the process. The rate of aerobic decomposition is generally quite rapid, with fresh plant litter losing as much as 50 to 70% of its original mass within the first year of decomposition. Below the water table, where oxygen is absent, the decomposition of organic matter is carried out by a succession of anaerobic microorganisms, each group utilizing a different electron acceptor to drive its metabolic processes. The first group to become active as oxygen is depleted is the nitrate-reducing bacteria, which use nitrate as their terminal electron acceptor, converting it to nitrogen gas or nitrous oxide in the process. When nitrate has been consumed, iron-reducing bacteria take over, using ferric iron as their electron acceptor and producing ferrous iron. Next in the sequence are the sulfate-reducing bacteria, which use sulfate as their electron acceptor and produce hydrogen sulfide, the gas responsible for the characteristic rotten egg smell that is sometimes noticeable near wetlands. Finally, when all other electron acceptors have been exhausted, methane-producing archaea, known as methanogens, become the dominant metabolizers, producing methane from the fermentation of simple organic compounds such as acetate and hydrogen. This sequential utilization of electron acceptors, known as the thermodynamic ladder or redox sequence, is a fundamental organizing principle of wetland biogeochemistry and is directly controlled by the hydrological conditions that determine the depth and duration of soil saturation. In a wetland with a permanently high water table, the redox sequence develops fully, with methanogenesis occurring at relatively shallow depths in the soil profile. In a wetland with a fluctuating water table, the redox sequence is repeatedly established and disrupted as the soil alternates between saturated and unsaturated conditions, leading to a more dynamic and spatially variable pattern of biogeochemical activity. The production and emission of methane from freshwater wetlands is a process of global significance that has attracted considerable research attention in recent decades, particularly in the context of climate change. Methane is a greenhouse gas that is approximately 20 times more effective than carbon dioxide at trapping heat in the atmosphere over a 100-year time horizon, and freshwater wetlands are the single largest natural source of methane emissions to the atmosphere. Global estimates of wetland methane emissions have been refined over the years as more field measurements have become available and better models have been developed. And current estimates suggest that freshwater wetlands emit between 170 and 260 teragrams of methane per year, with a best estimate of approximately 200 teragrams per year. The rate of methane emission from a particular wetland depends on a complex interplay of factors, including the temperature of the soil, the depth of the water table, the availability of organic substrates for methanogenesis, the presence of alternative electron acceptors such as sulfate that can suppress methanogenesis, and the efficiency of methane oxidation in the aerobic zone above the water table. Temperature is one of the most important controls on methane production, with rates typically increasing by a factor of 2 to 3 for every 10 degrees Celsius increase in temperature, following the general pattern described by the Arrhenius equation. This temperature sensitivity means that methane emissions from wetlands tend to be highest during the warm summer months and lowest during the winter. Though emissions can continue at significant rates even under ice cover in boreal and Arctic wetlands if the soil temperature remains above freezing. The pathways by which methane reaches the atmosphere from the anaerobic zone where it is produced include diffusion through the soil and water column, ebullition, which is the release of methane in the form of bubbles, and plant-mediated transport, in which methane travels through the parenchyma of wetland plants and is released to the atmosphere through the stomata or through breaks in the plant tissue. The relative importance of these three pathways varies among different types of wetlands and under different environmental conditions. Ebullition tends to be episodic and can release large quantities of methane in short bursts, particularly when drops in atmospheric pressure or disturbance of the sediment cause trapped gas bubbles to break free. Plant-mediated transport can be a major pathway in wetlands dominated by species with well-developed parenchyma, such as Phragmites australis or Typha latifolia. and can account for as much as 50 to 90% of total methane emissions in these systems. The carbon balance of freshwater wetlands, which encompasses both the uptake of carbon dioxide through photosynthesis and the release of carbon through decomposition, methane emission, and the export of dissolved organic carbon in outflowing water, is a critical factor in determining whether a wetland functions as a net source or sink of atmospheric carbon. Over long timescales, the accumulation of peat in wetlands represents a net transfer of carbon from the atmosphere to the terrestrial biosphere. And this peat accumulation has played a significant role in regulating atmospheric carbon dioxide concentrations over the past several thousand years. The total amount of carbon stored in the world's peatlands has been estimated at between 500 and 600 gigatons, which is approximately equivalent to the amount of carbon currently present in the atmosphere as carbon dioxide. This vast carbon store is vulnerable to release if peatlands are drained or otherwise disturbed, a possibility that has prompted calls for the conservation and restoration of peatlands as a strategy for mitigating climate change. The dissolved organic carbon that is exported from freshwater wetlands in their outflowing water is an important component of the carbon cycle and has significant effects on the chemistry and ecology of downstream water bodies. Dissolved organic carbon in wetland outflow is derived primarily from the partial decomposition of plant litter and peat and consists of a complex mixture of organic compounds, including humic and fulvic acids, sugars, amino acids, and other molecules. The concentration of dissolved organic carbon in water draining from freshwater wetlands typically ranges from 10 to 50 milligrams per liter, which is several times higher than the concentrations typically found in water draining from upland forests or grasslands. The brown or tea-colored appearance of many wetland-influenced streams and lakes is a direct consequence of the high concentrations of dissolved humic substances in the water, which absorb light in the blue and ultraviolet portions of the spectrum and impart a characteristic amber or brown color. The movement of water through the landscape, from hillslope to valley floor, from surface to subsurface, and from wetland to stream, is a continuous and interconnected process that cannot be fully understood by examining any one component in isolation. The concept of the hydrological catchment, which is the area of land from which all precipitation drains to a common outlet, provides a framework for understanding how wetlands fit into the broader landscape hydrological cycle. Within a catchment, freshwater wetlands typically occupy the lowest-lying areas where water accumulates, either in topographic depressions, along the margins of rivers and streams, or in areas where the water table reaches the surface. The position of a wetland within the catchment influences the sources and amounts of water it receives, the chemical composition of that water, and the role it plays in regulating downstream flows. Headwater wetlands, which are located in the upper portions of a catchment near the drainage divide, tend to be small and receive most of their water from precipitation and shallow groundwater seepage. These wetlands often have relatively simple water budgets and are particularly sensitive to changes in precipitation patterns because they have no large upstream water source to buffer them against variations in rainfall. Despite their small individual size, headwater wetlands can collectively occupy a significant proportion of the total wetland area within a catchment and can have a substantial influence on downstream water quality and flow regimes. Research in the Appalachian Mountains of the eastern United States has shown that headwater wetlands can reduce peak storm flows by 20 to 40% and can remove significant quantities of nitrogen and phosphorus from water before it reaches downstream channels. Floodplain wetlands, which are located along the margins of rivers and are periodically inundated by floodwaters, have a very different hydrological character than headwater wetlands. These systems receive large inputs of water during flood events, when the river overflows its banks and spreads across the adjacent floodplain, and they gradually drain back into the river as floodwaters recede. The frequency and duration of flooding in a floodplain wetland depend on the size and flashiness of the river, the elevation of the floodplain relative to the river channel, and the presence or absence of levees or other flood control structures. In a natural, unmodified river system, the floodplain may be inundated several times per year during high-flow events, with each flood lasting from a few days to several weeks. The sediment and nutrients deposited on the floodplain during these floods contribute to the high productivity of floodplain wetlands and make them among the most biologically diverse and ecologically important habitats in the landscape. The hydrological restoration of degraded freshwater wetlands has become an important area of applied science and environmental management in recent decades, driven by growing recognition of the many ecosystem services that wetlands provide and the extensive losses of wetland area that have occurred as a result of human activities. It has been estimated that approximately 50% of the world's wetlands have been lost since 1900, with the majority of these losses attributable to drainage for agriculture, urban development, and other land uses. In the conterminous United States, the rate of wetland loss was particularly severe during the mid-20th century, when federal agricultural policies actively encouraged the drainage of wetlands, and an estimated 53% of the original wetland area had been converted to other land uses by the 1980s. The restoration of hydrology is widely regarded as the single most important step in wetland restoration because the hydrological conditions of a site determine the soil conditions, nutrient cycling processes, and plant communities that can develop there. In many cases, the degradation of a wetland began with some form of hydrological modification, such as the construction of drainage ditches, the installation of subsurface drain tiles, or the diversion of surface water away from the wetland. Restoring the hydrology of such a site typically involves reversing or mitigating these modifications, for example by blocking drainage ditches, removing drain tiles, or reconnecting the site with its natural water sources. The success of hydrological restoration can be assessed by monitoring the water table depth and hydroperiod of the restored site and comparing these measurements with reference data from intact, undisturbed wetlands of the same type. The design and construction of constructed wetlands for water treatment purposes represents another important application of wetland hydrological principles. Constructed wetlands are engineered systems that are designed to mimic the water purification functions of natural wetlands and are used to treat a variety of water types, including municipal wastewater, stormwater runoff, agricultural drainage, and industrial effluent. The design of a constructed wetland involves careful consideration of the hydraulic loading rate, which must be sufficient to achieve the desired level of treatment without overloading the system, the vegetation to be planted, which influences both the aesthetic appearance and the treatment efficiency of the system, and the configuration of the flow path, which determines the distribution of water within the wetland and the contact time between the water and the treatment media. Two main types of constructed wetlands are in common use: surface flow wetlands, in which water flows across the surface of a shallow, vegetated basin, and subsurface flow wetlands, in which water flows through a bed of gravel or other porous media below the surface. Surface flow wetlands more closely resemble natural wetlands in appearance and function, and are generally used for the treatment of relatively clean water, such as stormwater runoff, or the polishing of effluent that has already undergone primary or secondary treatment. Subsurface flow wetlands are more commonly used for the treatment of more heavily polluted water, such as raw or primary treated municipal wastewater, because the subsurface flow path provides greater contact between the water and the microbial communities that carry out the treatment processes, and also prevents the exposure of the public to potentially hazardous wastewater. The hydraulic design of a constructed wetland typically aims to achieve a uniform distribution of flow across the entire area of the wetland, with a minimum of short-circuiting, which is the tendency for water to follow preferential pathways through the system rather than spreading evenly across its full extent. Short-circuiting reduces the effective treatment area of the wetland and can result in some of the incoming water passing through the system without adequate treatment. Various design features can be incorporated to minimize short-circuiting, including the use of elongated, narrow cells that force the water to follow a long flow path, the installation of baffles or berms within the wetland to redirect flow, and the use of multiple inlet and outlet structures to distribute flow more evenly. The residence time of water in a constructed wetland, which is the average length of time that a parcel of water spends in the system, is a key design parameter that is calculated by dividing the volume of water in the wetland by the volumetric flow rate of the incoming water. The long-term performance of constructed wetlands has been the subject of extensive monitoring and research, and the results have generally been encouraging. Studies conducted at operational constructed wetlands around the world have shown that these systems can achieve high levels of removal for many common water pollutants, including suspended solids, biochemical oxygen demand, nitrogen, phosphorus, and pathogenic microorganisms. Typical removal efficiencies for total suspended solids range from 80 to 95%, while removal efficiencies for biochemical oxygen demand typically range from 70 to 90%. Nitrogen removal, which relies primarily on the microbiological processes of nitrification and denitrification, typically ranges from 40 to 70%. Though higher removal efficiencies can be achieved in systems that are specifically designed to promote these processes by providing alternating aerobic and anaerobic zones. The study of wetland hydrology has benefited enormously from advances in remote sensing technology, which have made it possible to monitor wetland water levels, extent, and vegetation conditions over large areas and at frequent time intervals. Satellite-based sensors, such as those carried on the Landsat, Sentinel, and MODIS platforms, can detect the presence of surface water by measuring the reflectance of electromagnetic radiation from the Earth's surface at various wavelengths. Water surfaces have a characteristically low reflectance in the near-infrared and shortwave infrared portions of the spectrum compared to vegetation and bare soil, and this difference can be exploited to map the extent of inundation in wetlands and other water bodies. The spatial resolution of these satellite sensors ranges from 10 meters for the Sentinel, 2 sensor to 250 meters for the MODIS sensor, and their temporal resolution ranges from daily for MODIS to approximately 5 days for Sentinel. 2, allowing the seasonal dynamics of wetland inundation to be tracked with unprecedented detail. Synthetic aperture radar, which is an active remote sensing technology that emits its own electromagnetic radiation in the microwave portion of the spectrum and measures the radiation that is reflected back from the Earth's surface, has proven to be particularly useful for monitoring wetlands because microwave radiation can penetrate cloud cover and vegetation canopies that are opaque to optical sensors. This ability to see through clouds is particularly valuable in tropical regions, where persistent cloud cover can severely limit the utility of optical satellite imagery. Radar backscatter from flooded wetlands has a distinctive signature that can be used to identify areas of inundation even beneath a dense tree canopy, making radar an invaluable tool for mapping the extent of swamp forests and other forested wetlands that are difficult to survey using optical remote sensing. The integration of field measurements, remote sensing data, and hydrological models has enabled scientists to develop increasingly sophisticated understanding of the water cycle in freshwater wetlands at scales ranging from individual wetlands to entire continents. Hydrological models that simulate the movement of water through the landscape, including the storage and release of water by wetlands, have become important tools for predicting the effects of land use change and climate change on wetland hydrology and for evaluating the potential benefits of wetland conservation and restoration. These models range in complexity from simple spreadsheet-based water balance models that can be applied to individual wetlands to complex, spatially distributed models that simulate the hydrology of entire river basins and require high-performance computing resources to run. The future of wetland hydrology as a scientific discipline is likely to be shaped by several emerging trends, including the increasing availability of high-resolution remote sensing data, the development of new sensor technologies for in situ monitoring of wetland conditions, The growing use of machine learning and artificial intelligence techniques for analyzing large hydrological datasets, and the pressing need to understand and predict the effects of climate change on wetland water resources. As the global climate continues to warm and precipitation patterns continue to shift, the water cycle in freshwater wetlands will inevitably be affected, and the consequences for biodiversity, water quality, carbon storage, and flood risk will be felt across the landscape. The continued study of these systems, using the full range of tools and techniques available to modern science, will be essential for informing the management decisions that will determine the fate of the world's remaining freshwater wetlands in the decades and centuries to come. The gentle, persistent movement of water through these quiet, unassuming landscapes continues as it has for millennia, each molecule following its own unhurried path from sky to soil, from root to leaf, from stream to sea and back again. The wetland receives and releases, stores and transforms in a rhythm that is at once ancient and ongoing, a rhythm that will continue long after the last page of the last scientific paper has been written about it. For the water cycle in freshwater wetlands is, at its most fundamental level, simply the earth doing what it has always done, moving water from one place to another in an endless, unspectacular, and profoundly important cycle that sustains all life upon this planet. The sediment dynamics within freshwater wetlands are intricately connected to the water cycle and deserve careful examination in their own right, for it is through the transport, deposition, and resuspension of sediment particles that wetlands perform some of their most important functions in the landscape. When water-carrying suspended sediment enters a wetland, whether from surface runoff, a flooding river, or an inflowing stream, the dramatic reduction in flow velocity that occurs as the water spreads across the broad, shallow, vegetation-choked expanse of the wetland causes the suspended particles to settle out of the water column and accumulate on the wetland floor. The rate at which particles settle depends on their size, shape, and density, with larger, denser particles settling more rapidly than smaller, lighter ones. Sand particles, which typically have diameters greater than 62.5 micrometers, settle out of the water column within minutes to hours after entering a wetland, while silt particles, with diameters between 2 and 62.5 micrometers, may remain in suspension for hours to days, and clay particles, with diameters less than 2 micrometers, can remain in suspension for days to weeks unless they are caused to aggregate and settle by chemical or biological processes. The total amount of sediment that is trapped by a freshwater wetland over the course of a year can be quite substantial. Measurements at research wetlands in various parts of the world have shown annual sediment accumulation rates ranging from less than 1 millimeter per year in isolated bogs that receive minimal sediment input to more than 10 centimeters per year in floodplain wetlands that are regularly inundated by sediment-laden floodwaters. The sediment that accumulates in a wetland is not merely an inert deposit, but is an active participant in the biogeochemical processes occurring within the wetland, serving as a substrate for microbial communities, a reservoir of nutrients and contaminants, and a medium through which water and dissolved substances move. The relationship between water depth and plant community composition in freshwater wetlands has been the subject of extensive research, and a general pattern has emerged that is remarkably consistent across different geographic regions and wetland types. In the shallowest zones of a wetland, where water depth typically ranges from 0 to about 15 centimeters, the vegetation is often dominated by sedges, grasses, and other low-growing plants that are adapted to conditions of intermittent flooding and exposure. As water depth increases to between 15 and 50 centimeters, taller emergent species such as cattails, bulrushes, and reeds become dominant, their robust stems and extensive root systems well suited to the challenge of growing in deeper water. In areas where water depth exceeds about 50 centimeters, emergent vegetation gives way to floating leaf species such as water lilies and pondweeds, whose leaves rest on or near the water surface, while their roots are anchored in the sediment below. In the deepest zones of a wetland, where water depth may exceed 1 meter, submergent species that grow entirely below the water surface, such as various species of Myriophyllum, Ceratophyllum, and Alodia, predominate. This zonation of vegetation along the water depth gradient is a direct consequence of the water cycle and the hydroperiod of the wetland, and changes in the hydrological regime can cause dramatic shifts in the distribution and abundance of different plant species. A prolonged increase in water depth, resulting from a period of above-average precipitation or a change in the management of upstream water resources, can cause the dieback of emergent species that are unable to tolerate the deeper water and their replacement by floating-leaved or submergent species. Conversely, a prolonged decrease in water depth can allow emergent species to colonize areas that were previously too deep for them, potentially converting open water areas to dense stands of emergent vegetation. These vegetation shifts can, in turn, affect the hydrology of the wetland by altering the resistance to flow, the rate of evapotranspiration, and the rate of organic matter accumulation, creating complex feedback loops between hydrology and vegetation that can be difficult to predict. The role of beaver dams in shaping the hydrology of freshwater wetlands is a topic that has received increasing attention from ecologists and hydrologists, particularly in North America and Europe, where beaver populations have recovered significantly from historic lows following centuries of intensive trapping. Beavers, which are among the most proficient natural engineers in the animal kingdom, construct dams across streams and rivers using logs, branches, mud, and stones, creating ponds that can range in size from a few square meters to several hectares. These beaver ponds function as small wetlands, storing water behind the dam and creating areas of shallow, slow-moving water that support dense growths of wetland vegetation. The hydrological effects of beaver dams extend well beyond the immediate vicinity of the pond, influencing groundwater levels, stream flow patterns, and sediment dynamics in the surrounding landscape. Research conducted at beaver dam sites in the Rocky Mountains, the Appalachian Mountains, and various European river systems has demonstrated that beaver dams can raise the water table in adjacent floodplains by as much as 40 to 60 centimeters, extending the area of saturated soils well beyond the margins of the beaver pond itself. This raising of the water table can transform dry upland areas into functioning wetlands, with all the associated changes in vegetation, soil chemistry, and biogeochemical cycling. Beaver dams also reduce the velocity of stream flow, which promotes the deposition of fine sediments and organic matter upstream of the dam, gradually filling in the pond and creating the flat, organic-rich valley bottoms known as beaver meadows that are a characteristic feature of many mountain landscapes in North America. The influence of tidal cycles on the hydrology of freshwater wetlands that are located near the coast is another important aspect of wetland water dynamics that warrants consideration. In estuarine environments, where freshwater rivers meet the salt water of the sea, the daily rise and fall of the tide can exert a significant influence on water levels in adjacent freshwater wetlands, even in areas that are located well upstream of the zone of saltwater intrusion. Tidal freshwater wetlands, which are defined as wetlands that experience regular tidal fluctuations in water level, but are not significantly affected by salt water, occur in the upper reaches of estuaries and in the lower portions of rivers that are subject to tidal influence. The tidal range in these systems is typically smaller than in the adjacent estuary, often amounting to only a few centimeters to a few tens of centimeters. But even these small fluctuations can have important effects on the ecology and biogeochemistry of the wetland. The twice-daily rise and fall of the tide in a tidal freshwater wetland creates a pulsing pattern of inundation and drainage that is fundamentally different from the seasonal pattern of water level fluctuation that characterizes most non-tidal freshwater wetlands. During each flood tide, water from the adjacent river or estuary backs up into the wetland, raising water levels and temporarily inundating areas of the wetland that are normally above water. During each ebb tide, the water drains back out of the wetland, lowering water levels and exposing these same areas to the atmosphere. This regular tidal flushing helps to maintain well-oxygenated conditions in the surface layers of the wetland soil, which in turn promotes the decomposition of organic matter and the cycling of nutrients. The tidal exchange of water also facilitates the movement of organisms, including fish, invertebrates, and larvae, between the wetland and adjacent aquatic habitats, contributing to the high biological productivity and diversity that is characteristic of tidal freshwater wetlands. The isotopic composition of water in freshwater wetlands provides valuable information about the sources and pathways of water through these systems, and has become an increasingly important tool in wetland hydrological research. Water molecules that contain the heavier isotopes of hydrogen, deuterium, and oxygen, oxygen 18, behave slightly differently from molecules that contain the lighter isotopes during phase changes, such as evaporation and condensation. When water evaporates from a wetland surface, The lighter isotopes preferentially escape into the vapor phase, leaving the remaining liquid water enriched in the heavier isotopes. By measuring the ratios of deuterium to hydrogen and oxygen-18 to oxygen-16 in samples of wetland water, precipitation, groundwater, and stream water, hydrologists can distinguish between water from different sources and estimate the relative contributions of each source to the wetland water budget. Isotopic studies have been particularly useful for investigating the water budgets of isolated wetlands that have no visible surface water connections to other water bodies. In these systems, it can be difficult to determine whether the wetland is sustained primarily by precipitation, groundwater, or some combination of the two, because neither water source can be directly observed or measured. However, the isotopic signatures of precipitation and groundwater are typically quite different, with precipitation having an isotopic composition that falls along the global meteoric waterline. A well-defined relationship between the deuterium and oxygen-18 ratios of precipitation at different locations around the world, and groundwater having an isotopic composition that reflects the average composition of the precipitation that recharged the aquifer, modified by any fractionation that occurred during infiltration and transport. By comparing the isotopic composition of the wetland water with those of precipitation and groundwater, it is possible to estimate the mixing ratio of these two sources and thereby determine the relative importance of each in the wetland water budget. The acoustic properties of freshwater wetlands, while perhaps not immediately associated with the water cycle, are in fact closely related to the hydrological conditions prevailing in these systems and have been shown to have important ecological implications. The sounds produced by water flowing through a wetland, including the gentle gurgling of water passing through dense vegetation, the quiet dripping of water from overhanging branches, and the soft lapping of small waves against the shoreline, create an acoustic environment that is distinctly different from that of adjacent upland habitats. These sounds provide auditory cues that are used by many wetland-dependent animal species for navigation, communication, and habitat selection. Frogs and toads, for example, rely heavily on acoustic communication during the breeding season, and the chorus of calling males that is a familiar feature of springtime in temperate wetlands is a direct consequence of the seasonal flooding that triggers breeding activity. The thermal buffering capacity of freshwater wetlands, which arises from the high specific heat capacity of water and the insulating properties of saturated organic soils, helps to moderate temperature extremes in the local environment. During the summer, the evaporation of water from the wetland surface and the transpiration of water by wetland plants consume large amounts of thermal energy, cooling the air above the wetland and creating a local microclimate that is several degrees cooler than the surrounding upland areas. During the winter, the large volume of water stored in the wetland and in the saturated soil releases stored heat slowly, helping to maintain temperatures above freezing in the root zone even when air temperatures drop well below zero. This thermal buffering effect has important implications for the survival of wetland organisms, many of which are adapted to the relatively stable temperature conditions provided by the wetland environment. The economic valuation of the hydrological services provided by freshwater wetlands has been the subject of considerable research in recent decades, as economists and environmental scientists have sought to quantify the monetary value of the benefits that wetlands provide to human societies. These hydrological services include flood mitigation, water quality improvement, groundwater recharge, streamflow regulation, and the provision of water for agricultural, industrial, and domestic use. Various methods have been used to estimate the economic value of these services, including the replacement cost method, which estimates the cost of constructing engineered alternatives to the natural services provided by wetlands, the avoided damage method, which estimates the cost of the flood damage or water treatment that would be required in the absence of wetlands, and the contingent valuation method, which uses surveys to estimate the amount that people would be willing to pay to preserve wetland services. The results of these economic valuations have generally indicated that the hydrological services provided by freshwater wetlands are worth substantial sums of money. A comprehensive global assessment published in 2014 estimated that the flood regulation services of inland wetlands were worth an average of approximately $2,500 per hectare per year, while the water purification services were worth an average of approximately $1,800 per hectare per year. When all ecosystem services are considered, including not only hydrological services, but also carbon sequestration, biodiversity habitat, recreation, and cultural values, the total economic value of freshwater wetlands has been estimated at between $10,000 and $40,000 per hectare per year, making wetlands among the most economically valuable ecosystems on Earth on a per-unit area basis. The management of water resources in landscapes that contain freshwater wetlands requires careful consideration of the complex hydrological interactions between wetlands, rivers, aquifers, and the surrounding land. Water management decisions that are made with the intention of benefiting one component of the hydrological system can have unintended consequences for other components, including wetlands. For example, the construction of a dam on a river upstream of a floodplain wetland can reduce the frequency and magnitude of flooding, which may benefit human communities living on the floodplain, but can also deprive the wetland of the periodic inundation that it requires to maintain its ecological character. Similarly, the extraction of groundwater from an aquifer that is hydraulically connected to a wetland can lower the water table beneath the wetland, potentially causing it to dry out even if the pumping well is located some distance away. The recognition of these complex hydrological interactions has led to the development of integrated water resource management approaches that seek to balance the needs of human water users with the maintenance of healthy wetland ecosystems. These approaches typically involve the establishment of environmental flow requirements, which specify the quantity, timing, and quality of water flows that are needed to sustain the ecological functions of rivers and wetlands, and the incorporation of these requirements into water allocation decisions. In Australia, for example, the Murray-Darling Basin Plan, which was adopted in 2012, allocates a specific volume of water for environmental flows in the basin's rivers and wetlands, ensuring that the water needs of these ecosystems are considered alongside the needs of irrigated agriculture, urban water supply, and other human uses. The monitoring of wetland hydrology using automated data logging systems has become standard practice at research and management sites around the world and has greatly improved the temporal resolution and quality of hydrological data available for these systems. Modern data loggers can be programmed to record water level, temperature, electrical conductivity, dissolved oxygen, and other water quality parameters at intervals ranging from minutes to hours, providing continuous records that capture the rapid fluctuations in hydrological conditions that can occur during storm events, tidal cycles, and other short-duration phenomena. These continuous records have revealed aspects of wetland hydrology that were invisible to earlier generations of researchers who relied on manual measurements taken at weekly or monthly intervals, including the rapid rise and fall of water levels during storm events, the diurnal fluctuations in water level caused by evapotranspiration during the daytime, and the complex interactions between surface water and groundwater that occur during the transition between wet and dry seasons. The diurnal fluctuation of the water table in a vegetated wetland during the growing season is a particularly interesting phenomenon that illustrates the close coupling between plant physiology and wetland hydrology. On a typical summer day, the water table in a densely vegetated wetland will begin to decline in the morning as the stomata of the plants open and transpiration commences. The decline continues throughout the day, reaching its maximum in the late afternoon when transpiration rates are highest. After sunset, the stomata close and transpiration ceases, allowing the water table to recover during the night as groundwater seeps in from the surrounding area to replace the water that was lost to transpiration during the day. The magnitude of this diurnal water table fluctuation typically ranges from 1 to 5 centimeters, depending on the density of the vegetation and the rate of transpiration. And this regular daily cycle can be clearly observed in continuous water level records from most vegetated wetlands during the growing season. The study of paleohydrology, which is the reconstruction of past hydrological conditions using evidence preserved in geological and biological archives, has provided valuable insights into the long-term variability of the water cycle in freshwater wetlands. Peat cores extracted from bogs and fens contain a wealth of information about past hydrological conditions, encoded in the types and abundances of plant remains, the humification of the peat, the concentration and isotopic composition of various chemical elements, and the preserved remains of testate amoebae, which are microscopic single-celled organisms whose species composition is closely related to the moisture conditions of the peat surface. By analyzing these indicators in dated peat sequences, researchers can reconstruct the history of water table fluctuations in a peatland over periods of hundreds to thousands of years, providing a long-term context for understanding current hydrological conditions and predicting future changes. Paleohydrological reconstructions from peatlands in the British Isles, Scandinavia, and other parts of Europe have revealed that the water tables in these systems have fluctuated significantly over the past several thousand years in response to changes in climate. During cool, wet periods, such as the Little Ice Age of approximately 1400 to 1800 of the Common Era, water tables in many European peatlands were relatively high, and the peatlands were actively accumulating peat at rates that were higher than the long-term average. During warm, dry periods, such as the medieval warm period of approximately 900 to 1300 of the Common Era, water tables were lower and peat accumulation rates declined. These findings demonstrate that the hydrology of freshwater wetlands is sensitive to climatic variability on timescales of centuries to millennia and suggest that the climate changes projected for the coming century are likely to have significant effects on wetland water budgets and the ecosystem services they provide. The quiet persistence of water moving through freshwater wetlands, seeping through layers of peat accumulated over thousands of years, rising and falling with the seasons, evaporating under the summer sun and replenishing with the autumn rains, represents one of the most understated yet vital processes in the natural world. Each droplet that enters a wetland embarks on a journey that may take days, weeks, months, or even years to complete, passing through root zones where it is absorbed and transpired by plants, filtering through soil layers where its chemical composition is gradually transformed by microbial activity, and eventually emerging as outflow to a stream, as vapor rising into the atmosphere, or as deep percolation to an underground aquifer. This unhurried passage of water through the wetland ecosystem is the fundamental process upon which all other wetland functions depend, from the storage of carbon in accumulating peat to the provision of habitat for countless species of plants, animals, and microorganisms that have evolved to thrive in the wet, quiet, endlessly cycling world of the freshwater wetland. The capillary fringe, which is the zone of soil immediately above the water table where water is drawn upward by capillary forces, plays an important but often overlooked role in the hydrology of freshwater wetlands. In fine-textured soils such as clays and silts, the capillary fringe can extend as much as 1 to 2 meters above the water table, maintaining near saturated conditions in the root zone even when the water table itself has dropped well below the soil surface. In the organic soils that are characteristic of many wetlands, the capillary fringe is typically between 10 and 50 centimeters thick, depending on the degree of decomposition and the pore size distribution of the peat. This capillary zone serves as a buffer between fully saturated conditions below the water table and unsaturated conditions above, providing a transitional environment that is exploited by a variety of plant and microbial species that are adapted to conditions of intermediate moisture. The electrical conductivity of wetland water, which is a measure of the total concentration of dissolved ions, provides a simple but informative indicator of the sources and pathways of water through a wetland. Rainwater, which contains very few dissolved minerals, typically has an electrical conductivity of less than 50 microsiemens per centimeter. Groundwater that has been in prolonged contact with mineral-bearing rocks and soils has correspondingly higher conductivities, often ranging from 200 to over 1,000 microsiemens per centimeter, depending on the geology of the aquifer. By measuring the electrical conductivity of water at various locations within a wetland and comparing these measurements with the conductivities of the potential source waters, it is possible to identify the zones where groundwater discharge is occurring, where precipitation is the dominant water source, and where mixing between different water sources is taking place. The concept of wetland connectivity has been extended in recent years to encompass not only the physical movement of water between wetlands and other water bodies, but also the ecological connectivity that is mediated by organisms that move between wetlands and the surrounding landscape. Many species of birds, mammals, reptiles, amphibians, and invertebrates use freshwater wetlands during some or all of their life cycle and move between individual wetlands or between wetlands and other habitats in the course of their daily, seasonal, or annual movements. These organism-mediated connections are important for maintaining genetic diversity within wetland populations, for the dispersal of plant seeds and other propagules between wetlands, and for the transfer of nutrients and energy between wetlands and the surrounding terrestrial and aquatic ecosystems. The water that moves through a freshwater wetland carries with it not only dissolved chemicals and suspended sediments, but also a vast and largely invisible community of microorganisms, including bacteria, archaea, fungi, algae, protozoa, and viruses. These microorganisms are transported passively by the flowing water and are deposited throughout the wetland as the water infiltrates into the soil, passes through vegetation, or pools in quiet backwaters. The microbial communities that develop in different zones of the wetland are shaped by the local hydrological and chemical conditions, with aerobic organisms dominating in the oxygenated surface waters and anaerobic organisms dominating in the saturated subsurface. The total abundance of microorganisms in wetland water and soil is staggering, with typical bacterial concentrations of 1 million to 1 billion cells per milliliter of water and 1 billion to 10 billion cells per gram of soil. The freeze-thaw cycles that occur in the soils of freshwater wetlands in cold climates have significant effects on the hydrology of these systems. When the water in the pore spaces of the soil freezes, it expands by approximately 9%, disrupting the soil structure and creating new cracks and macropores that can alter the hydraulic conductivity of the soil when it thaws. Repeated freeze-thaw cycles can significantly increase the porosity and permeability of the upper soil layers, facilitating the infiltration of snowmelt water in the spring and potentially increasing the rate of groundwater recharge. In peatlands, the freezing and thawing of the peat can cause the surface to heave upward during winter and subside during spring, a phenomenon that is known as frost heave and that can displace individual peat hummocks by as much as 5 to 10 centimeters vertically over the course of a single winter. The influence of atmospheric pressure changes on water levels in freshwater wetlands is a subtle but measurable phenomenon that has been documented at a number of monitoring sites. When atmospheric pressure decreases, as typically occurs ahead of an approaching low-pressure weather system, the reduced weight of the atmosphere above the wetland allows the water surface to rise slightly, and the water table in the surrounding soil responds similarly. Conversely, when atmospheric pressure increases, the added weight of the atmosphere pushes down on the water surface, causing both the surface water level and the water table to decline slightly. The magnitude of these pressure-induced water level changes is generally quite small, typically amounting to only a few millimeters for a change in atmospheric pressure of 1 millibar. But they can be clearly detected in high-resolution water level records and must be taken into account when interpreting these records. The relationship between wetland hydrology and the global carbon cycle is one of the most important and actively researched topics in environmental science today. Freshwater wetlands occupy a unique position in the global carbon budget, functioning simultaneously as sinks of atmospheric carbon dioxide through the accumulation of peat and as sources of atmospheric methane through the anaerobic decomposition of organic matter. The net effect of these opposing fluxes on the global climate depends on the relative magnitudes of the carbon dioxide uptake and the methane emission, as well as on the different atmospheric lifetimes and radiative forcing efficiencies of these two greenhouse gases. Over short timescales of decades to centuries, the warming effect of methane emissions from a growing peatland may exceed the cooling effect of carbon dioxide uptake, making the peatland a net contributor to global warming. Over longer timescales of millennia, however, the accumulated carbon dioxide uptake eventually outweighs the methane warming effect, and the peatland becomes a net climate cooling agent. The partitioning of water between different flow paths in a wetland catchment is influenced by the antecedent moisture conditions of the soil, which refers to the degree to which the soil is already wet or dry before a rainfall event begins. When the soil is dry, a large proportion of the incoming rainfall is absorbed by the soil matrix, replenishing the depleted moisture stores and contributing to the slow, steady flow of water through the subsurface. When the soil is already wet or saturated, as is often the case in the contributing area of a wetland, the soil can absorb little additional water, and a much larger proportion of the rainfall runs off across the surface, entering the wetland quickly and causing a rapid rise in water level. This dependence of runoff generation on antecedent moisture conditions means that the hydrological response of a wetland to a given rainfall event can vary enormously depending on whether the event occurs during a wet period. When the soil is already near saturation, or during a dry period, when substantial moisture deficits exist in the soil. The nutrient spiraling concept, which was originally developed to describe nutrient dynamics in streams and rivers, has been applied to freshwater wetlands to describe the downstream movement and cycling of nutrients as they are carried through the wetland by the flowing water. As a nutrient molecule such as a dissolved phosphate ion moves through the wetland, it may be taken up from the water by a plant or microorganism, incorporated into living biomass, released back into the water upon the death and decomposition of the organism, taken up again by another organism, and so on, tracing a spiraling path downstream as it alternates between dissolved and particulate forms. The length of each spiral, known as the spiraling length, is a measure of the efficiency with which the wetland retains and recycles nutrients, with shorter spiraling lengths indicating more efficient retention. In densely vegetated wetlands with slow flow velocities, spiraling lengths for phosphorus can be as short as 10 to 50 meters, indicating that a phosphorus molecule is likely to be taken up and released several times within a relatively short distance. The water that eventually leaves a freshwater wetland, whether by surface outflow, groundwater seepage, or evapotranspiration, has been profoundly altered by its passage through the system. The physical, chemical, and biological processes that operate within the wetland have removed suspended sediments, transformed dissolved nutrients, degraded organic pollutants, and modulated the flow regime, all through the simple mechanism of slowing the movement of water and allowing time for these processes to occur. This transformation of water quality and flow is the essence of the ecosystem services that freshwater wetlands provide to human societies, and it is entirely dependent on the maintenance of the hydrological conditions that allow these processes to function. The protection and restoration of freshwater wetland hydrology is therefore not merely an ecological imperative, but an economic and social one as well, for the services that these systems provide freely and indefinitely, if left intact, would cost billions of dollars to replicate using engineered alternatives. As the hours pass and the water continues its unhurried journey through the wetland, rising imperceptibly through the capillary fringe, filtering through the dense root mats of sedges and cattails, pulling in shallow depressions where dragonfly larvae hunt among the submerged stems, and seeping slowly toward the outlet where it will rejoin the stream and continue its long journey to the sea, the great and ancient cycle continues. The wetland breathes water in and breathes it out again, a slow and steady respiration that has sustained these landscapes through ice ages and warm periods, through droughts and deluges, through millennia of change and continuity. And in this unhurried breathing, in this patient cycling of water from sky to earth and back again, there is a quietness and a constancy that speaks to something fundamental about the nature of the world, something that has no need of explanation or excitement, something that simply is and always has been and always will be, as long as rain falls and water flows and the earth receives what the sky provides.